Today we're going to look at the physiology of pain in a kind of a nuts and bolts pain receptor connected to noxious neuron type pain. This is the definition of pain given by the International Association for the Study of Pain, and it's the definition that is becoming more and more widely accepted. I would say that whenever I see somebody put up a slide with a definition of pain on it, 9 times out of 10 this is the one that they use. If you look on the website, you will see that it comes with about 3 pages of footnotes which are explanatory, but the basis of it is that pain is an unpleasant sensory and emotional experience. It's unpleasant. If it's not unpleasant, it's not pain. And it is both a sensory and an emotional experience. It has 2 aspects to it. We perceive pain in the environment, and pain has a certain emotional effect on us. This emotive aspect to pain contributes to make pain the least objective and the most subjective of the senses. What I mean by that is that if you look at this fire extinguisher here, it's a red fire extinguisher. Okay, your sense of colour says that's red. And your sense of colour is fairly objective and fairly robust. And when I've conducted experiments on this, I can tell you that after at least 8 pints of good ale, it's still red. Okay? You can alter the way your central nervous system works, but the objective redness of something that's red doesn't change. If you think of pain, pain is much more subjective, and it has been said that the degree of pain that one perceives is due more to the state of the central nervous system that's perceiving it than due to the actual magnitude of the stimulus. And that is something that we will explore. Coming down to the second part of the definition, we're talking about pain being associated with tissue damage or described in terms of such damage, and talking about it being a complex perception. The described in terms of tissue damage is important because until relatively recently it was believed that any organism that couldn't describe its pain couldn't perceive pain. And so the concept of pain stopped at the point at which children developed language, and a pre-linguistic child was not believed to be capable of perceiving pain, to the extent that it was routine for such very young children to go through fairly major surgery without being given analgesia, because they couldn't perceive pain. Glad to say that that position has moved now, and moved significantly, to the extent that we believe that all humans after birth have the ability to perceive pain, and also most vertebrates fall into a class of animal that most people will allow to perceive pain at most points of their life. However, this descriptive terminology does lead to some interesting points and causes some problems. The medical practitioners found out about 20-25 years ago now, to some of their surprise, that if you want to know how much pain a patient is in, the best way to find out is to ask them. And a lot of practitioners spent a long time looking for an objective measure that correlated well with that pain. But the best measure you can get is asking the patient. That leaves us with a bit of a problem in terms of asking a dog how much pain it's in. Woof, not the most useful response. But also, for our medical colleagues, the problem of groups of human patients where verbal communication is not possible. Again, the pre-language children and patients with cognitive impairment who have difficulty in communicating. It's very difficult to find out how much pain those patients are in. And so we have to think very carefully about how we assess pain, how we approach pain, particularly in our patients as veterinarians. I've also said that pain is a complex perception that takes place in the higher centres of the nervous system. And we know that but we also know that pain in some instances can get from our periphery to those higher centres. And so we have to talk about the pathways by which those impulses travel up and become pain. To allow us to do that, we use the term nociception. Nociception is a stimulus which would be perceived as painful if it reached a conscious central nervous system. So if you imagine an animal that's under general anaesthesia but doesn't have any specific analgesics in it, when we perform surgery on it, those action potentials go up the sensory nerves, get to the spinal cord, go up the spinal cord to the midbrain, but then they begin to be attenuated and they don't reach the higher centres of the central nervous system, so the animal doesn't feel pain. We talk about all those neurons and all those pathways below the level of conscious perception as nociceptive. So a noxious stimulus stimulates a nociceptor and leads to the generation of action potentials in nociceptive pathways. And that's an important distinction, certainly to have in your mind in the next lecture when we get on to talking about the effects of pain and the ways in which we can provide analgesia. There are 4 distinct types of pain, and they are each quite different entities. Pinprick pain, also known as fast pain. It's pain that's carried in the A fibres, and it has certain characteristics. First of all, yes, it's definitely pain, but it's a mild sensation of pain. It's well localized. You can tell where it's coming from. It doesn't have major psychological issues, and it doesn't persist. The perception of pain stops as soon as the stimulus stops. So that's pinprick pain or fast pain. Let's go on to talk about acute pain. It's different to fast pain, and yet it's the same. First of all, it's definitely pain. We identify it as pain. But it gets to us more slowly. Yeah, you feel that you've banged your hand, and you think, ooh, that'll hurt in a minute, and lo and behold, it does. It radiates. On a cold morning, if you bang your thumb with a hammer, it can go to your elbow, it can go to your shoulder. It's not localized to the place where the insult was. It can alter your behavior, it can make you vocalize, it can make you walk around in small circles with your hand tucked under your arm. It changes the way you behave. And the sensation persists beyond the stimulus. You don't bang your thumb with a hammer and go, ooh, that was painful, never mind, and get on with your life. The sensation of pain persists. And so that is acute pain. And that's C-fibre pain, mediated through the small and myelinated fibres and through the reticular formation. The next sort of pain, in most of the books you will see termed as chronic pain, but it's more correctly termed persistent pain, and in some of the more recent reviews and texts it's beginning to be called persistent brackets chronic pain, and I guess soon it will just be persistent pain. It's pain which persists beyond your expectations. Bang your thumb with a hammer and you have a kind of expectation that that's going to be sore for a while. Maybe for a few hours, maybe the next day. But if it's sore in 3 weeks' time, then that has persisted beyond your expectation. And so it's persistent pain. And persistent pain is completely different from acute pain in the sense that it doesn't have very much to do with the stimulus, but it has a lot more to do with the central nervous system. Given a patient's history, and particularly their history of painful conditions and exposure to previous painful insults, they can perceive or are likely to perceive pain as more long-lasting or less long-lasting. And often, when we see animals that are in persistent pain, there isn't very much in the way of a lesion to find. The best example I can give you for that is lower back pain in humans. Lots of people suffer from lower back pain as they get older. In many people it's debilitating and it stops them living their life in the way they want to. And yet when you look at these patients, it's very, very difficult, in fact impossible in many cases, to pinpoint an abnormality, to pinpoint a lesion. You can't see anything abnormal that's causing that pain. That pain is a feature of an abnormality in the spinal cord, in the pain processing pathways. It's a sensitization of those pathways, it's an aberrant response in those pathways, which is causing that pain to persist beyond everybody's expectations. And when you get onto pain therapy, you'll find that chronic pain needs to be treated in very different ways to acute pain. Analgesics that are really good for acute pain seem to be very much less good for persistent pain, and vice versa. The last form of pain is the most unpleasant, and that's neuropathic pain. Neuropathic pain is pain due to a lesion or disease process in the central nervous system itself. Might be in the spinal cord, in the dorsal horn, might be more centrally in the midbrain, might be in the cerebral cortex. Might even be to some extent in the peripheral nervous system. And an example of that is a pain called phantom limb pain, which is pain that patients suffer sometime after they've had a limb amputated. So if you imagine somebody has this arm amputated, classically 9 to 18 months later they will go back reporting pain in this hand that doesn't exist anymore. We call it phantom limb pain, and it can't be due to a lesion in the hand because it's gone. It's due to changes in the central nervous system, changes in the way that that system is working and interpreting its environment. As we go down from pinprick pain to neuropathic pain, the pain is getting more severe, more pathological, more unpleasant, and also getting harder to treat. So that if you've got pinprick pain, you don't go screaming to the hospital demanding opioids. And if you did, then very mild analgesics will probably sort you nicely. Neuropathic pain can be sufficiently depressing that it completely stops you functioning in your normal life. And yet it's extremely difficult to treat properly, or even at all in some patients. So we're moving down through a spectrum of 4 very different things that we're calling pain, but they're not all the same thing, and it's not appropriate to treat them all in the same way. Okay, let's go on to talk about the history of pain and what we know about pain perception in a physiological sense. The first person that had anything to say about pain in a physiological sense was Descartes. And some 400-500 years ago, Descartes' view of pain was summed up in this diagram. He envisaged a nervous system as a collection of tubes, small tubes which ran from the periphery up to the brain, where they connected with the ventricular system of the brain and transmitted information to the pineal gland. Now that's an interesting theory by modern standards, but it's actually a very appropriate theory for the time. Descartes was working just after the invention and development of the optical microscope, and using the best optical equipment available in his day, a section of a nerve fibre would visualize the myelin, but not really very much else. And so those nerves looked like, guess what, a collection of hollow tubes. And so this hollow tube explanation was fairly logical. Electricity was unknown at the time, and so if you've got hollow tubes that look fairly robust, wouldn't it make sense that the way to get information up and down them is to move fluid through them? A sort of hydrostatic mechanism, well known to science, and many of the most modern machines in his time employed that kind of technology. The pineal gland sticks out from the brainstem into the ventricular system. So wouldn't it make sense that something that was kind of sticking out into this fluid system and maybe movable when fluid moved was something that was responding to those sensations? And so Descartes developed this theory of the nervous transmission of pain going from the periphery and being perceived centrally. And with one or two small changes, and allowing for the discovery that nerves worked electrically by Voltaire and others about 200 years later, this theory of transmission of nervous impulses from the bottom to the top with perception at the top persisted right until the middle of the last century. Not bad to get a 400-year theory in medicine. The big change that went on in the middle of the last century was the beginnings of an understanding that pain could be processed at different levels. You'll notice here that there's no hierarchical processing. These tubes go right to the top and the processing goes on at the top. In the last century, workers moved the theory on to allow for processing within the spinal cord. This is the work of Meltsack and Wall, published in 1965, possibly the most famous diagram in the study of pain, referred to as the gate theory diagram. So what's happening here? Well, Meltsack and Wall and others had realised that different sensations can affect the perception of pain. Bang your thumb with the hammer, and one of the things that you do is you rub it. Or if you're a dog and it's sore, you lick it. Yeah, you manipulate the area around it. If you've got a child that's hurt themselves, you offer to rub it better for them. And they realised that that really does reduce the perception of pain. And so they needed to incorporate into what they knew about nociception a mechanism by which different sensory perceptions could alter the perception of pain. So they had the concept of an inhibitory interneuron, which sat in the dorsal horn of the spinal cord and integrated the signal where the C fibre and the A beta fibre went up to the central nervous system. So you can see here that we've got mechanoreceptor fibres coming in and stimulating neurons which are going on to the reticular formation in the central nervous system, and we've got C fibres coming on and stimulating those same neurons. But here we've got an inhibitory interneuron which is inhibiting these two. That interneuron is itself inhibited by the C fibre, and so the C fibre is actually amplifying the pain or noxious stimulus that's going on to the central nervous system. But it's stimulated by this A beta fibre. And so when this A beta fibre is stimulated, we get perception going on to the central nervous system, but this inhibitory interneuron is stimulated, and so the perception is to some degree reduced. And this interneuron can be stimulated by the A fibre and inhibit the C fibre, which allows us to look at the way in which mechanical stimulation can reduce the perception of pain. Importantly, it introduced two concepts into the study of pain and analgesia. One was the concept of plasticity, the fact that the spinal cord can undergo changes at these synapses and learn to be more sensitive or less sensitive to noxious stimuli, and so more or less likely to transmit those stimuli onto the higher centres to be perceived as pain. Secondly, it gave us a place in the spinal cord for the opioids to have an analgesic action. And as I'll show you towards the end of the lecture, this interneuron has become the encephalinergic interneuron, a neuron which responds to opioids and other analgesics. What I want to do now is to look at plasticity and look at the ways in which the perception of pain or this perception of noxious stimuli can be altered by different things going on in the animal. This is a Great Dane, or rather a small part of the flank of a Great Dane, backside. Let's orient ourselves. We've got an anus here and a tail going on that way. We've got a hind limb coming down here, and we've got a head somewhere outside over there. This is a Great Dane that came into a veterinary practice to be spayed, to have an ovohysterectomy. And indeed it was spayed. And because it was such a big dog, and there wasn't really room to spread it out properly in the kennels, it was decided to recover it on the floor in the anaesthetic induction room. Good plan. On a nice comfy blanket. Good plan. Next to a radiator. Less cunning. This lesion here, which when it was referred to us for surgical treatment, was 5 centimetres long, 3 centimetres wide and 2 centimetres deep, is the necrotic hole left when the burn that dog got from the radiator had sloughed. So what happened was, because the dog was still anaesthetised and it was next to a hot pipe of a radiator, it couldn't behaviourally adjust things, it couldn't move away from the radiator, and by the time it was able to do that, all this tissue had been killed. And the dog was very painful when it came to us. Those of you that have had burns will know that burns are extremely painful things. And the reason is that these necrotic areas of tissue here spit out molecules which sensitize C fibres. So, for example, potassium, the major intracellular cation. You lyse the cells, the potassium leaks out into the extracellular fluid, and guess what? It sensitizes these bare nerve endings on the C fibres. Prostaglandins, manufactured by enzymes that exist in the extracellular space from cell wall debris, from arachidonic acid, sensitize nerve endings. Bradykinins, histamine. Every time I look at this literature, there are more of these molecules which are released into damaged tissue and which sensitize nerve endings. As a group, we call them eicosanoids, and last time I looked, there was something in excess of 5,000 of them. Lots and lots of them. But basically, damaged tissue sensitizes the nerve endings in the intact tissue around them and increases the perception of pain. One of the best things you can do for a lesion like that is to debride it and to get rid of that source of eicosanoid, that source of damaged tissue glue or soup or whatever you want to call it. And that will make the patient significantly more comfortable. So we've got sensitization within the peripheral tissues. Let's look now at how those pain pathways get to the central nervous system. And the good news is you've already seen some of this. So the somatic nerve endings, those in the body, travel with the somatic nerves. So nothing new to learn about there. And from the viscera, as we found last semester, the pain pathways travel with the autonomic nerves. And you'll remember that they are segmental. So that here we have autonomic pain pathways travelling with parasympathetic nerves, caudal to the bladder, travelling with sympathetic nerves between the bladder and the heart, travelling with parasympathetic nerves cranial to the heart. The same sort of segmental basis as the outflow of the autonomic nervous system, or a similar segmental basis. And that's interesting clinically, because when we look at animals that are having surgery, usually when we give them a noxious stimulus, they get a tachycardia, a hypertension. They get a response to noxious stimulation that looks like a sympathetic stimulation. But occasionally they get the opposite. Occasionally we see bradycardia, hypotension. And sometimes it can be quite a sudden, not usually devastating, but certainly impressive and exciting hypotension and bradycardia. And the areas of the body which are more likely to cause that bradycardic response seem to be those areas caudal to the bladder and those areas cranial to the heart. So that laryngeal surgery, tracheal surgery, and surgery of the reproductive tract and the rectum are all prime candidates for causing this bradycardia hypotension response. And it's not been demonstrated, but it may be that there is some kind of overlap between the sensory pathways travelling with these parts of the autonomic nervous system and the response to noxious stimulation that we see. But now we've got a model for plasticity in tissues and an understanding of how those nerve fibres get to the spinal cord, get to the central nervous system. And once they get to the central nervous system, there is even more opportunity for plasticity and for alterations in that response. Here we've got information coming from the receptor. Feeding into lamina 10, or the spinal reticular formation. And ascending to the higher centres of the central nervous system in the spino-reticular tract. Coming from the spinal cord, going to the reticular formation, the spino-reticular tract. But this pathway is very different from the pathways that we looked at in the last lecture with the other bodily sensations. And the difference is in the spreading out of the response. So that here we've got responses going into the spinal reticular tract, going up a few segments, and going back into the reticular formation, a few more synapses, coming back out, going up. The whole thing is a branching, diverse network. And I've heard it described as like an onion bag or a hairnet, a sort of a diverse network of fibres travelling in lots of different directions. Travelling bilaterally, going all the way up, going some of the way up, stopping on the way. And that explains two of the things which we see when we look at acute pain. First of all, it explains this radiating, spreading out, poorly localized nature, in the sense that whenever these synapses occur, there is a reverberation in the signal. And so as the signal ascends the central nervous system, it spreads out. It becomes greater in magnitude, it becomes less precise. And it explains the slowness of the response. Remember that the peripheral fibres are unmyelinated C fibres, so the information is getting from the periphery more slowly than the other senses anyway, and it's taking this non-direct route through the central nervous system. It's spreading out in the central nervous system, and it's affecting different places. And it gives us a very potent, very powerful, yet slightly slower system of sensation. When we get to the cranial reticular formation, again I've put on here similar segments to the segments we saw last time with the sensory nerves. We've got a medulla, a midbrain, and this time separately a thalamus. The reticular formation is still in the centre of the central nervous system, periaqueductal, right through to the central thalamic nuclei, and possibly, although the jury is still out on this, into the cerebral cortices. And again, you can see that we've still got this piecemeal, bilateral, spreading out, slow pathway with other senses feeding into it. This is the auditory sense coming from the colliculi. Again, a sound can be so loud that it's painful, or so unpleasant that it's painful, and for different people that's different sounds. For me, that's nails scraping down a blackboard. Ooh. So just thinking about it is actually quite unpleasant. But when I hear it, as I try and avoid doing most of the time, it's a painful sound. It really is nasty. So different senses can feed into the reticular formation and feed into this sensation of pain. Now, had I been lecturing you on pain, where are we, 2007, 12 years ago probably, this is where I'd have stopped, at the midbrain and the thalamus. And I would have said that the perception of pain is the business of the structures within the midbrain, the limbic structure, or limbic system as it was then more commonly called. And that the cerebral cortex was incidental to the perception of pain. However, there was a lot of work done in the mid-90s using what were then very new imaging techniques which looked at responses to pain in both volunteers and pain patients in the cerebral cortices. This is an example of one of these studies. This is a functional MRI study of 2 sections through the brain of a volunteer who is sitting in an MRI scanner with his hand on a plate. And the plate is arranged so that fairly rapidly it can be made warm or cold. When the plate's made warm, various areas in the sensory cortex start to become active, to interpret that warmness, to sort out the sensation, to work out what's going on, to add that warmth in the hand to the volunteer's picture of the world around him. But when that heat becomes noxious, when it starts to get hot and painful, then 2 particular centres, the anterior cingulate gyrus and the insular cortex, become active. And those centres weren't active for warmth. They become active when the volunteer starts to report pain. Similarly, when we cool the plate down, we get activity here in the sensory cortex, but nothing much else going on. And when that cold becomes noxious, we start suddenly to get activity in the anterior cingulate gyrus and also in the insular cortex. So we've got here 2 specific areas of the cerebral cortex which are not specific for the sensory modality, but they are specific for when that sensation becomes sufficiently intense that it's painful. And the sensation of pain could be a pressure pain, could be thermal pain, could be chemical pain. It doesn't matter on the actual modality of the pain. These centres respond to something that is painful no matter what sort of pain it is. And subsequent work has shown us that particularly the anterior cingulate gyrus seems to be involved in a decision about a perception. So that when we perceive something as a sensory input, the anterior cingulate gyrus says, is that painful? And if it decides it's not painful, then that thing becomes perceived as a sensation. And if it makes a decision and says, no, that is painful, then it changes the way we perceive that thing, it changes the way that perception alters the central nervous system, and it becomes painful. So pain is being looked on less and less as a perception, and more as a decision that the central nervous system makes about the world around it. We get to decide on what we perceive in terms of whether we think it's painful or whether we think it's not painful. And these studies, together with other studies that have followed them, have really turned on its head the way we think about pain as a sensory modality, and also begun to heavily influence the way that we treat pain. Let's go back to gait control and plasticity in the dorsal horn. And I said to you that this neuron is now considered to be the enkephalnergic interneuron. So I just want to put that into a more anatomically correct location. And here it is in the dorsal horn of the spinal cord, the enkephalnergic interneuron. And these are all the pathways which can affect the enkephalnergic interneuron and can affect pain perception. First of all, in yellow, we've got true afferent pain, C-fibre pain, coming through to the periaqueductal area, the central area of the grey matter, ascending in the spinoreticular tract. We've got a touch afferent in blue, well, kind of grey-blue, coming in here, ascending to the higher centres of the central nervous system in the dorsal funiculus, and here inhibiting the true pain pathway, the C-fibre pathway. Here we've got a pinprick afferent, again ascending bilaterally and acting via the enkephalnergic interneuron to affect C-fibre pain. And so this purple and the yellow and the red here are the components of the diagram that I showed you previously. And also in green we've got the descending reticulospinal system, synapsing with the enkephalnergic interneuron, and sometimes called the diffuse noxious inhibitory system, the DNIS. But this is a pathway that's coming down, a motor pathway in a sense, from the higher centres of the central nervous system to influence the amount of pain sensation or noxious sensation that ascends from this segment of the spinal cord. And this system allows us to control the amount of pain that we perceive. This descending reticulospinal system can be affected by all sorts of things in our patients, by motivation, by environment, by depression. In persistent pain patients, depression very often shuts down this descending reticulospinal system and makes the pain that they perceive more severe, which makes the depression more severe, which shuts the system down even more, and so we get a circulating vicious cycle of pain perception getting worse and worse. And often we can give antidepressant drugs which will contribute to the patient's well-being and reduce the patient's pain. Not because they're analgesic in themselves, but because they take away the depression that's reducing this descending reticulospinal system. I just want to show you now a diagram which looks in a time sense at that plasticity of pain and looks at how our perception changes after a noxious stimulus. So you can see that we've got from a millisecond time frame here up to days, weeks and months at this end, and these are all the ways in which the pain pathways alter over time in response to a noxious stimulus. So in the first few milliseconds we've got AMPA receptors and then NMDA receptors and second messengers all within the cells altering the cell's responsiveness to a noxious stimulus. Later on, we've got the development of neurokinins and immediate early genes, which begin to alter the expression of protein on the cell surface, change the receptors, change the number of receptors, so that patients with persistent pain have a different population of sodium receptors on their neuronal membranes than healthy patients. And those different populations of sodium receptors respond in different ways to different analgesics. Later on, we've got growth factors and immunomodulators, an increase and then decrease in receptor numbers, possibly neuronal remodeling. The central nervous system is learning to interpret the pain. It's altering its synapses, it's changing its structure to interpret this pain. And then, depending on the magnitude of the pain, after hours, days, weeks, even months, we get the return of normal sensation. So whenever we get a noxious stimulus, we're influencing a whole cascade of mediators and mechanisms which are altering the way our central nervous system works and altering the way our central nervous system perceives that noxiousness as pain. And we'll get on to looking at that in more detail in the next lecture.